Thanks, Orlando. It's my pleasure to be presenting to you Theme 1. My name is Lindsay Eltis. Our theme is focused on the biocatalytic transformation and engineering of biomass, ultimately to valorize it. This theme includes researchers from really three different faculties and many different departments. So I think it's really symbolic of the cross-cutting interdisciplinary work that gets done at the BPI. And the researchers in this theme are really cover the gamut of understanding biological systems that are both responsible for synthesizing biomass, plants, for example, and breaking it down, so microorganisms, fungi, and bacteria. And the researchers focus on from the very small level, looking at individual genes, proteins, enzymes, how they function in these complex biological systems, and then also applying systems biology to understand how these different components interact with each other. Finally, we're applying things like systems biology to try and engineer these systems in order to help valorize the biomass. So I'll just walk you through a couple of the highlights of some of the research that these individuals have accomplished over the last year. So I'll start with Sean Mansfield, and he's been doing some really cool work to engineer different kinds of lignin that plants can synthesize. So he's been doing this using a variety of approaches, but one of the sort of more innovative things he's been doing is taking bacterial genes and inserting these into plants so that they make different kinds of lignin. So for instance, introducing ester bonds into the lignin so that it's easier to break down and not only do more interesting things with the lignin itself, but also to more easily access the other components of the biomass. And another thing that he's doing is, again, through engineering the plants with bacterial genes and enzymes, he's able to introduce new chemical moieties that decorate the lignin so that these can be harvested as platform chemicals. Then there's Lacey Samuels, also in the Department of Botany. She's been looking at different systems in plants that biosynthesize not just lignin, but also plant secondary metabolites such as cannabinoids. She's done some very innovative work on understanding how lignin is synthesized, or rather how the monolignals that are used to biosynthesize the lignin are diffuse out of the cells without any transporters, and this diffusion is driven by the polymerization of the lignin outside of the cell, which therefore generates a concentration gradient. Interestingly, cannabinoids itself as well, just like lignin, are synthesized outside of the plant cell, and she's identified a organelle, a trichrome, that is very lipid rich and is responsible for this, drives this biosynthesis through a very high lipid flux. I skipped one over there somehow. Oh yeah. And then sorry, this thing skips around a lot. My own group is involved in the bacterial degradation of lignin, and both understanding how this occurs and then manipulating these systems in order to affect the use biocatalysis to help valorize the lignin. We study both pathways within these microorganisms that are responsible for degrading lignin-derived aromatic compounds, and also identifying rate-limiting steps in this process. So identifying the enzymes that have to be engineered or manipulated in order to engineer these systems. So one of the very interesting characteristics of these pathways is that very different kinds of lignin-derived aromatic compounds all get basically catabolized in a similar manner. So this sort of facilitates our ability to engineer these systems in order to convert a wide array of aromatic compounds to single bioproducts that can be used as platform chemicals, things like adipate that are a building block for nylon. So we've, again, collaborating with a number of other researchers in the BPI, we've been able to identify novel pathways, and maybe I'll talk a little bit more about that shortly. Another researcher in the Department of Microbiology and Immunology here in this building actually is Bill Moen, and he's also interested not only in the bacterial catabolism of these lignin-derived aromatic compounds, but also the ecology of the microorganisms that are responsible for this degradation. He's helped develop a number of innovative techniques that allow us to, or allowing him to identify novel biomass degrading organisms and novel genes involved in this process. And he's doing this using not only metatranscriptomic and metagenomic approaches, but also techniques such as stable isotope probing. And he's applying these techniques to very interesting ecosystems here in British Columbia, such as the Liard Hot Springs, where there's biomass being degraded at elevated temperatures. So when he goes bioprospecting for enzymes and genes in this environment, he can find enzymes that are thermostable, for example. Again, very useful for biocatalytic applications. So I just want to take a moment to highlight one of the papers that we came out with this year, again, sort of because it exemplifies, I think, some of the cross-cutting collaborative work that gets done within the BPI. And this involved a collaboration between myself, Bill Moen, Scott Reneker, and Stephen Withers. And this was involved in, this paper was aimed at identifying a novel or the degradation pathway for a compound that had never been described before. But really the motivation for this work stemmed from the fact that some novel approaches have been developed for valorizing lignin, because that's something that's really critical to do if we're going to make next generation biorefining sustainable. And so this is really the integration of chemocatalytic and biocatalytic approaches in order to extract more value from the lignin. So essentially deconstructing the lignin into a variety of monoaromatic compounds, so using a chemocatalytic process, and then twinning this with a biocatalytic approach where this mixture of aromatic compounds that's being generated is then converted using an engineered microorganism, so a microbial cell factory, to convert that mixture into a single bioproduct that can then be used. And so one of the chemocatalytic approaches that's being developed in this approach is oxidative catalytic fractionation. And when that's applied to biomass, it not only gives you a fraction of cellulose and carboxylic acids, but it also breaks down the lignin. So you get predominantly vanillin and acetovinilone in amongst the monophenols, as well as a variety of smaller lignin oligomers. So some of these monophenols, if they're going to be valorized biocatalytically, you actually have to know the enzymes and the pathways that are responsible for that valorization. And one of these compounds is acetovinilone, whose degradation has never been described before. A student in Bill's lab and a postdoc in mine were able to isolate a bacterium from the UBC farm compost here, actually, that was able to grow on acetovinilone. They identified the genes that were responsible for this, and then Stephen Withers' group, a graduate student in that group, synthesized some of these intermediates of this pathway so that we were able to use them to validate the pathway. And then we showed that the organism that we'd isolated was able to grow on a mixture of aromatic compounds generated by oxidative catalytic fractionation. And that oxidative catalytic fractionation was done in Scott's lab. So this was like a really nice collaboration. And now that we have these genes, we're able to use them to engineer microorganisms to degrade this compound along with others to convert them into these platform chemicals. And these are using genetic tools that we and others are developing for modifying these enzymes. So this really nicely illustrates just the collaboration within the BPI, but also the whole range of different techniques that we're using. So then just to finish off with to highlight the contributions of a couple of other researchers in theme 1 then. There's Jörg Bollmann over in the Michael Smith labs, and he's interested in plant bioproducts and forest genomics. So he's identifying genes and pathways responsible for interesting plant secondary metabolites. This paper came out recently where he had identified the pathway responsible for the biosynthesis of an anti-diabetic plant metabolite. And he's also involved in genome sequencing of some of the trees that occur in the Pacific Northwest, such as the western red cedar, and getting insights into the low genetic diversity of this particular species. And finally, Harry Brummer is also at the in the Michael Smith lab. He's a chemist primarily. He works on different carbohydrate active enzymes, and doing a lot of very interesting work on some of these lytic polysaccharide monooxygenases and related copper radical oxidases. And he's been able both to engineer these enzymes in order to alter their activities. So again, very important for biocatalytic applications. But he's also been able to take these enzymes in order to chemically functionalize cellulose, nano, and macrofibers, so liberating carboxylic acid so that for subsequent chemical modification, and really being able to then develop these biocatalysts for the functionalization of cellulosic materials. So I think I'll leave it there, and I hope this has given you a really good overview of the kinds of research that gets done in theme 1, and also the huge variety of PIs that we have contributing to this theme. So thanks.